Brilliant. Okay, thanks very much. Yes, so this project, I'm the data scientist, and it's with Sean Bayliss from the vet school, and Axel Montu, who's in engineering. He's the person who did the work, in some sense. And the problem that we were working on is, well, let's look at this tree in the bottom left. So we've got some pathogens, right? They could be bacteria, they could be viruses, whatever. These pathogens are sitting in some database somewhere, and they're ones that we've observed. And we're imagining, when a new sequence comes in, for the majority of the time, they will be like in this top group here, they will be very similar to something that we've seen previously, and that's because most of these pathogens reproduce clonally. So there will just be some small change from something that we've seen, and so we'll be able to predict its function very easily. And so there's many of these clonal groups. So here's another one down here. The problem comes from a kind of prediction of what function these organisms will have when you get something like the red one, right, that is not very similar to anything that we've previously seen. And why might it not be very similar? So one explanation is that it is a distant relative of everything in our current database. So maybe it's a rare pathogen, or maybe it infects a different sort of species normally. And in that case, we might not know what its function's going to be, but it's quite unlikely to be effective in infecting humans. Normally, these sorts of infections don't work out for the organism. So that's one scenario. But another scenario is that it doesn't look like this species because it's actually recombined its DNA with this other one, right? So these sort of hybridization events are the sexy side of phylogenetics, and they're a little tricky to detect because they're really the same, overall average genetic distance from everything in our sample as in both cases. So what we've done is develop a pipeline that does that. So it takes the genome of bacteria, so that we've got the A species here and we've got the B species here, and if those two species are together in one individual, they've co-infected one individual, then one of them can choose to take up DNA from the other. And if they do that, they can also take on the functional properties of the other. And so this is exactly how antimicrobial resistance genes can spread across bacterial types and other sorts of nasty pathogenic behavior can transfer to. What our algorithm does is it goes through the genome and it just checks for every one of these like long branches, does it look like this has happened in a quick way. And so that's where we are currently. We've got this technology, it's built. What we're quite excited about is the idea of next taking this and putting it on a server somewhere so that it will just watch these databases as they're built, and it will maintain this tree, and it will look at the tree and say, is this branch actually due to recombination or not? And then people will be able to subscribe to notifications and say, has a particular gene been recombined into this organism, and so on. So that's the next steps for this, and please talk to Sean or myself if you're interested. Thanks very much.
Great, thank you. Do we have any questions for Dan? Yeah, let's go Ben and then you, Roberta.
Thanks, Dan. That was very interesting. I have a question around more kind of flipping it the other way, looking at where some of these pathogens, obviously, you need to have a susceptibility that it's attacking, right? Is there a way that you can almost look at it from let's call it the animal, the human, looking at where, as obviously we evolve or genetically mutate ourselves, where there are going to be new potential vectors that we can get infected by that weren't previously there, if you see what I'm saying?
So obviously there's a constant stream of new infections coming from animal populations. Of course, humans don't tend to adapt very quickly compared to the timescale that the pathogens do. So recently, that doesn't, that way round hasn't happened. But historically it has. You can see it in the genome, right? Early farmers brought with them a whole bunch of diseases, and then they moved and infected hunter-gatherers with those diseases, and they had no immunity to it, and so that rapidly changed what humanity was resistant to.
Thank you. That's very interesting. I just want to know what is the impact or what impact you foresee from this research. For example, who are going to be the main users of the technology you are developing? And also, how can it help either prevent or tackle antimicrobial resistance?
So the obvious thing that you can do with it is just basic science, right? So you can understand which lineages are recombining, which we don't really know, and that tells us about how the history of co-infection has occurred. So that sort of history is a kind of first stage in building up a really good understanding of what all these genes do, right? So that's one of the things that we want to do. The idea of getting these alerts to specific recombination events could be used at least by kind of second stage healthcare researchers who work directly with, for example, monitoring active threats. I wouldn't imagine those people are directly ready to ingest alerts that say, some gene's just been recombined. But as part of an alert system, I think that, in 10 years' time, I think we'll see a lot of this sort of thing.
Great. Any other questions? No? In which case, thanks very much, Dan.
Thank you very much.